Welcome to the Performance Around the Clock podcast. I am Professor Satchin Panda, a scientist in the field of circadian rhythm. In this podcast, I will talk to experts in many different fields about science, health, and human performance. You'll also find out why circadian rhythms is my favorite topic. I hope you enjoy the podcast and thank you for listening. Welcome to another episode of Performance Around the Clock, and I'm super excited to have a esteemed professor, Professor Joe Takahashi, with me. In modern days, when you're talking about circadian rhythm, sleep, metabolism, behavior, disease, it's very hard to find a basic science discovery or paper that is not directly or indirectly unrelated to Joe Takahashi. So that's why I'm super excited. Welcome, Joe.
Thank you, Satchin.
Yeah, you have a very long, storied history, career in circadian rhythm research, starting from your PhD, I guess. And when I was working on circadian light receptor, how light affects circadian clock, I was kind of pleasantly surprised because that time I knew you as working on core circadian clock mechanism, but I was pleasantly surprised to see that the idea that blue light affects circadian clock much more efficiently than any other color of light actually came from your research. So can you tell us how you got into circadian rhythm?
Yeah, sure. So my PhD advisor was Michael Menaker, and he had discovered extraretinal photoreceptors in birds. And so we always wondered what the photopigment for that was. And then his lab started working on golden hamsters to study photoperiodic control of reproduction, which is light controlled. And we were studying the circadian properties of golden hamsters. And we were wondering what the properties of light were that effectively shifted the clock.
Yeah.
And so we were able to very quantitatively measure the response to light in golden hamsters using monochromatic wavelengths of light.
Very well-defined color of light.
Right. And, in vision, we don't talk about color, we talk about wavelength, and the active ingredient is actually the photon.
Yes.
And so what's important is how many photons are actually absorbed by a photopigment, not its wavelength.
Yeah.
And so we were motivated to try to understand what is the spectral sensitivity or the action spectrum for phase shifting the clock in a mammal. And so we undertook these experiments when I was a graduate student with Pat DeCourcy, who was a visiting scientist in the lab at the time, and another undergraduate student. The three of us did all these experiments together in hamsters. And what we found is that the action spectrum peaked actually in the close to the green.
Yeah.
But there is very clear blue or short wavelength sensitivity also. And when we measured the action spectrum, it didn't match any known photopigment in the hamster retina.
So this is the typical rod and cone photopigment.
Yeah. It peaked near the rod photopigment, which is rhodopsin, around 500. But we discovered a very interesting feature when we did these experiments, and that is that the hamster could integrate light over a very long period of time. So there's a trade-off between intensity and duration. And so for a rod photoreceptor, that time constant is measured in 1 or 2 seconds.
Yeah.
Okay? But for the clock response, it was measured in tens of minutes. And we actually showed that the hamster could integrate time for at least 45 minutes.
Wow.
Back then. And so that told us that this was a very unusual photoreceptive system.
So that also meant that if somebody flicks a light switch, maybe for 4 or 5 seconds to check what time it is in the middle of the night, that may not be sensed by the circadian system, but if the light is on for 15 to 20 minutes, then
No,
to say that or no?
no, it can still sense a very short light pulse, like a photographic flash. We
Yeah.
did those experiments too,
Yeah.
but it has to be much brighter.
Yeah, extremely bright.
Yeah. So right. So what we found is it's the number of photons in the light, and it could be over a very short period of time or a very long period of time.
So if somebody wants to reset the clock with a flash or very short period of light, then that has to be extremely bright.
Extremely bright.
But at the same time, even if there is dim light over 40-45 minutes, that can also be
That's correct.
effective.
Right.
Yeah.
Yeah, and the threshold that we found for long pulses is on the order of moonlight.
Wow.
Yeah.
So moonlight can still,
Yeah, full moonlight.
full moonlight,
Yeah.
of course. So that's around
3/10 of a lux.
3/10 of a lux.
Yeah.
Yeah.
Mhm.
So that's, so one lux is roughly one candle light.
That's right.
Couple of feet away from you, so it's 3/10. It's pretty dim, but that can still reset. So that might explain some of the lunar activities in some, like the owl monkeys are awake at night in full moon, and then they're asleep at night during new moon. So there are quite a few phenomena about lunar rhythm. So maybe this
Yeah,
kind
it
of
could be. Yeah,
could be, but we haven't conclusively seen that in any system.
that's right.
So then how, so that was retina photoreceptor, because hamster don't have extra retinal photoreceptor.
That's correct.
So from retina, then you moved into the brain to look at the circadian.
Yeah, eventually, many years later, we looked at how light, increases gene expression. It was really FOSS induction. This was in the 1990s.
Yeah.
Discovery made by Bill Schwartz's lab and Benji Brusek's lab.
So light activates this expression of CFOS, which is usually considered as a neuronal activation marker.
Right. Yeah.
Okay.
Yeah, so I had a really good graduate student who worked on that. His name was John Kornhauser. And one of the clear things that he showed was that the induction of FOSS, this is again in the hamster, was quantitatively correlated with the phase shifting response of the same light pulse. So we had this nice quantitative correlation. So that made us think that the FOSS response was relevant.
Yeah.
And then the other very surprising effect was the FOSS response was gated. It only occurred during the subjective night.
Yeah. So this is something that many people outside circadian rhythm don't understand. What is the gating of response? Maybe you can explain a little bit more. And which we see in many aspects of physiology and metabolism.
Yeah, it's really, you can think of it as a permissive state. At some phases of the cycle, you can get a response, and other phases you do not get a response. What was remarkable about this is even with a very bright light pulse during the subjective day, there is no FOSS response at all.
Yeah.
It's not like it's smaller.
Yeah. So that means even though the circadian clock is in the complete darkness, and that means the eye must be sensing a lot of light, still the clock prevented, the circadian system prevents activation of
Right.
the SCN.
Yeah.
Yeah.
And I would say it's still a mystery today. We don't understand that phenomenon.
Really?
Yeah.
Is the chromatin different for FOSS locus at different time of the day?
Yeah, we don't know.
Okay.
Yeah, I don't think it's going to be at that level.
So then you also worked on the pineal.
Yeah, so again, as a graduate student, I worked on developing a system that allowed us to measure circadian rhythms continuously from the chicken pineal in culture using the release of melatonin into the culture medium using a perfusion system where we collected fractions that were timed. And then that gave us beautiful circadian rhythms of melatonin release to show that the chicken pineal indeed had a circadian oscillator. And it was interesting because the chicken pineal is also photosensitive.
Yeah.
So we could entrain the oscillator in vitro.
Yeah.
And
No, this is important that chicken pineal is photosensitive, but not the mouse or human pineal.
that's correct.
Yeah. Because I hear once in a while people say, well, you got to shine light on your head because the pineal is photosensitive, which is untrue for humans and
That's
mice,
correct.
to be very clear. So was that then the chicken pineal experiment? So this is the first, this is an experiment where you, of course, you're sacrificing the chicken or taking out the pineal from the chicken and putting it in a dish. And this pineal now produces melatonin in every 24 hours. Was that, to your knowledge, the first example of ex vivo demonstration of circadian rhythm, or the SCN was done around that time too?
Good question. I think that, we show there are 4 cycles of melatonin. So I think back then, the SCN was really only measured through electrophysiology.
Yeah.
And it was, difficult to measure even 1 cycle of electrical activity in SCN.
Yeah.
So
This must be one of the very earliest, if not the earliest one.
in a vertebrate tissue.
Vertebrate tissue.
Yeah.
Yeah.
The other extremely good example back then was the eye of aplesia,
Yes.
right? I would say that was the best in vitro oscillator.
But in this case, chicken pineal, also in birds, melatonin plays a bigger role in synchrony. So
Right.
it's almost, I won't say with some qualification that chicken pineal may be as strong as the SCN is in mammals, because if you disrupt the melatonin rhythm in some birds, then they lose many rhythms.
Yeah, but it turns out only in some birds.
In some birds.
Passerine songbirds, the pineal is very important. But in a chicken or a quail,
It's not?
it's redundant.
Okay.
So the other piece of work I did as a graduate student was to lesion the suprachiasmatic nucleus of house sparrows. And that caused a loss of rhythm in house sparrows. So it showed that structure had a common function in both birds and mammals.
I see.
But in addition, there's the pineal gland.
Yeah.
And later, people show the retina had a clock in birds.
So then how did you go from back? So you started with hamster, did some chicken and house sparrow. How was your journey to mouse?
So that's many years later, my journey to mouse. So in between, I did a postdoc with Martin Zatz at NIMH, and we worked on the chicken pineal together. And then when I got a faculty position at Northwestern, I focused on the chicken pineal system, but we used dissociated or dispersed cells, primary cell cultures, instead of explant cultures of the gland.
Yeah.
And that made our system much more quantitative because we could use 96 well plates, have many more replicates.
So from one pineal, you can just dissociate the cells and you can put it in multiple wells.
That's correct, yeah. Or we'd take a group of pineals and make very large cultures.
So you have to kill less chicken.
That's correct, yeah. But the rhythms were much better in those cells because I think the cells had better culture conditions than an explant, which doesn't last that long. It maybe lasts a week and a half or 2 at the most. So using the dissociated pineal cell culture system, we did a lot of experiments trying to understand the input pathway, the output pathway, and also what the mechanism of the clock might be. And so one of the areas that we explored was the effect of protein synthesis inhibitors, which was essentially a conserved intervention. Every circadian system that had been tested with inhibitors of protein synthesis showed that that would cause very strong phase shifts and changes in period.
Which implied that there is transcription and translation.
That's right. So then I had a very talented MD PhD student. His name was Jose Flores, and he took on this project to look for cycling proteins. And so what he did is he used two-dimensional gel electrophoresis, radio-labeled proteins.
Wow, a lot of radioactivity.
Yeah, S35 methionine.
Yeah.
And we used this system that was developed by Jim Garrels at Cold Spring Harbor that allowed you to see about 1,000 spots on a large 2D gel.
That's a lot of, because I had tried once 2D gel. You can barely get, say, few dozen, but 1,000 spots is a lot.
Yeah, we could see close to 1,000 spots with the Garrels system. And so Jose, would take samples from these cell cultures over the circadian cycle, and he found half a dozen spots that had huge circadian rhythms of incorporation of S35 methionine. And so one of them was 56 kilodaltons, and he was then able to make preparative 2D gels, cut out the spot, and obtain about 3 micrograms of protein, which is a lot of protein. Okay, and we sent it off to the Harvard Peptide Sequencing Core. And they sent us back the peptide sequences, and it was very clear what the protein was. It was tryptophan hydroxylase, which was very disappointing to us because we already knew about tryptophan hydroxylase. Well, we didn't know it cycled, but we knew it was on the biosynthetic pathway for the synthesis of melatonin. It wasn't part of the clock. And so that for me was a watershed moment in my career, because here we had the most sophisticated proteomics technology at the time.
Yeah, that was in the 90s.
Yes. We could detect 1,000 spots. Okay. Maybe we could identify a handful of those if there was enough protein. Okay. Yet, if you thought about it, a cell expresses 10,000 proteins. We could only see the tip of the iceberg. Everything else was invisible to us. Okay. So when I came to that realization, I knew that this is not going to work. We can't see 90% of what we're looking for. We need a different way. And I came to realize that the only way you can find something that you don't know anything about is genetics. Okay, because you need no preconceived information on the nature of that protein. All you really need is a good, robust phenotypic effect. Okay. And so at that time, one of my faculty colleagues, his name was Larry Pinto, who worked on the visual system. He was using different mouse mutants in the retina to study vision. And he had found out that a colleague of ours at Wisconsin was using ENU mutagenesis in mice.
So it's a mutagen that is injected into the...
Yeah, so they were mutagenizing mice in Madison, Wisconsin, testing for a liver enzyme for phenylalanine hydroxylase.
So this is related to cancer project or something?
Distantly, no, they were studying this disorder, metabolic disorder. And so we met with them, teamed up, and agreed to collaborate. So they screened their mice that they'd mutagenize in Madison, and then they shipped them down to us in Evanston, Illinois. And then Martha Vitaturna, talented postdoc in my lab, then screened the mice for wheel running activity. And so Martha, well, during this time we had learned that the mutation rate for ENU could be as high as 1 in 700
genes
per locus per gamete. So that's 1 in 700 per gene per genome, if you want to translate it. That rate was for recessive mutations, not for dominant mutations. But that was the only rate that was available.
So that's extremely rare also.
It's not so bad.
1 in 700, so you still have to, it's not that bad. So I think a lot of people who don't know about wheel running phenotype, I think mice and many organisms now, we know if they are given access to a running wheel, like a hamster wheel, then they will run on this wheel 24/7. And even if you turn off the light, they will run with a very defined circadian period. So that means for these 6 mouse, in every 23 hours, 45 minutes, they will wake up and run. And since it's a very precise measurement, even if there is 10 or 15 minutes change, or even if you're lucky, 1 hour change in their clock, then you can find it.
Right. Yeah, so that rate, 1 in 700, meant that if we screened 3 times that number, 2,100 mice, we might cover the genome threefold if the rate was at that rate. And so I invested 100 running wheel channels to the screen, and the screen would take about a month, so we could screen 100 a month. In a year, we could screen 1,200. And in 2 years, we'd screen more than 2,100. So it seemed feasible. So that's what we did, okay? But we were even more lucky than that, because Martha put on the first batch of mice, which was about 40 mice that she tested. And mouse number 25 had a 1 hour long period, okay? Now, 1 hour seems subtle, but it was a it was a dominant screen, meaning only one chromosome was mutated.
So this is, to put in context, 1 hour is less than 5%.
That's correct.
Change in phenotype, which will be extremely hard to find in any other mouse behavior. For example, learning and memory.
Impossible.
Right. Grooming activity, or even a 5% change in metabolic rate may not be visible.
Or even an enzyme assay.
And that's the power of circadian rhythm phenotype. Even a few minutes change in our clock can be detected and quantitatively assessed. And
and the reason that worked is the variability of the period between mice was extremely small. It was less than, the standard deviation was less than 0.2 of an hour. So when we found the 1 hour period change, it turned out that that was 6 standard deviations away from the mean, or what we call a z-score of 6. Which is huge. Very strong. So that led to the discovery of this mutant. It took 2 more generations to make the mutation homozygous, because the first founder mouse was heterozygous.
Had only 1 copy of the mutation.
That's right. When we crossed that mouse, which was male, to wild-type females, almost half the progeny from that cross were mutant.
So that's
So we knew,
dominant.
yeah,
Sorry, yeah.
it was consistent with a single gene semi-dominant or dominant mutation. Okay, then we took two affected offspring from that cross and crossed them together to make an F2 cross to get the first possibility of a homozygous mutant. And again, we're kind of incredibly lucky. The very first litter had 4 mice, okay? By phenotype, 1 was wild type, 2 were heterozygous, and 1 had an even stronger phenotype.
So that's like classic Mendelian
That's right.
textbook.
Right, 1 to 2 to 1 ratio. And that mouse had a 28-hour period when we put it in darkness, and then after about 2 weeks, the rhythm went away.
Wow.
And so we were able to replicate, that phenotype in many other presumed homozygous mice.
So Joe, there are many things that we don't see in the manuscript. Do you remember the first time Martha told you about the mutant? What was your reaction? Or when you had the first generation F1, what was your reaction, or the F2?
Oh yeah, well I mean, when we first saw the mutants,
Yeah.
we were excited, but it was the first batch of mice. So we didn't really know whether that was going to be reliable. I think once we made that first cross and it was transmitted to the offspring, that was huge, because then we know it's genetically transmitted.
Yeah.
Okay. But, that's a long ways from understanding what it was. But then we eventually mapped the gene to chromosome 5, and we could show or prove it was caused by a single gene.
Yeah.
And so we knew that we had a mutant.
Yeah.
And interestingly, its phenotype is as strong as, the strongest Drosophila or fly mutants known at the time. 28-hour period.
Yeah.
This is about the same as the Perlong mutant.
Yeah. Any other mutant came out of that screen, or did you continue that screen after the first 40 mice?
Well, we screened about 300 mice and then we stopped. And
Because at that time it was a huge amount of effort to clone.
that's correct. Yeah. I think the other interesting part of that story is when we did the screen, everybody thought we're crazy. Because no one had ever done a behavioral screen using ENU in mouse. So there's that. And then the other, strong bias, which, you hear even today, is that complex phenotypes, which we thought circadian rhythms were complex, are rarely controlled by single genes.
Yeah.
Most, they think it's more like height or body weight, which is controlled by many interacting genes.
So Joe, at that time, it's back to what, 95, 96, that's when that time you were still young associate professor?
No, I was a full professor
You were already full professor, but you said that at that time, no one believed that ENU mutagenesis can be used to get a mutant in behavior. Did that imply that the funding agencies were also skeptical? How was the funding to?
Oh, we didn't try to get NIH funding for that. We had a pilot grant from the MacArthur network that gave us a very small pilot grant that allowed us to begin. And then in the early 90s, Gene Block had obtained an NSF Science and Technology Center in biological timing at the University of Virginia. And we were one of the 3 institutions part of that. And that center funding is actually what supported our work and our ability to clone the gene.
Yeah. But I guess that's also another example that sometimes, like you said, you figured out. I think somebody had told me that a sign of a good scientist is to figure out a dead alley before you are at the end of the dead alley. And I guess you figured out from the proteomics experiment that that's not a feasible way to figure out the mechanism or identify the components of the clock and go in a different direction.
Right.
And this is something that we see in many successful scientists, that they take a big chance, because again, this was a huge chance. At that time, no one was doing this kind of experiment, and you kind of went for it.
Yeah, but I think that, the original Konopka and Benzer paper in
Yeah.
Drosophila from 1971, that was always, my point of reference,
Yeah.
is that it was possible to do it in Drosophila, and then there's this spontaneous mutant in the hamster, which had a 20-hour period
Yeah.
caused by a single gene, and that said it can happen in a mammal.
I see.
So those two findings together convinced me that it can happen.
But then coming back to the cloning of this gene,
Yeah.
which is now, you named it as clock. Do you remember the day or any moment that was exciting that you...
Yeah, there are many moments in that. So, well, let me go back to the paper. When we published the paper on the mutant mouse, we tried to name the mouse Clock, but one of the reviewers said, you can't name it Clock because you don't know its function is associated with the clock.
I see.
So then we had to make up a new name, which is this very long name called Circadian Locomotor Output Cycles Kaput.
Yes.
And the acronym of that is CLOCK.
Yeah.
So that's how we got the name CLOCK.
Who came up with that name? Because it's a very smart name.
Oh, I did.
Okay. Yeah. Because you are essentially looking at the output of the clock, which
That's right.
is the activator of them. Yeah. So
That's right. So anyway, once we had the mutant, positional cloning was very non-trivial
yeah.
in the mid-90s.
Yeah.
The first human genes had only been cloned in the early 90s, and those early genes actually had chromosome abnormalities, which you could see
on gel sometimes, like RFLP and all this.
sometimes, yeah.
Sometimes.
Right. And so it was difficult. Probably the first human gene to be cloned that was a point mutation was cystic fibrosis.
Yeah.
And that took a huge effort at the time. So what we had to do first was to make a very high resolution genetic map. Martha Vitaterna again did the crosses, and David King, a graduate student, very talented, he essentially did all the genotyping for those crosses.
How many mice? It must be 2,000 plus mice.
Yeah, so in the end we used 2,600 meioses,
Okay.
which was a combination of backcross and intercross. So I don't know exactly how many mice.
Yeah.
And then when we had a very high resolution map position, there were no genes that had been identified in that region, and there weren't many markers. We had already used up all the genetic markers in that region. And so then we had to switch to physical mapping, which is to isolate the DNA from that region. And so back then they had what are called YACs, or yeast artificial chromosomes, which have very large inserts, megabase size inserts. And we did find a megabase YAC clone that covered our region. But YAC clones are very hard to work with because they're low copy number, and they're very unstable. And the libraries were unreliable because
They would recombine.
yeah, many of them had recombined genomic segments from two regions of the genome. So at the time, I was lucky. I was introduced to Jeff Friedman at Rockefeller by Mike Young, who had invited me to visit Rockefeller.
Yeah, Jeff Friedman, by the way, who discovered leptin and
An
leptin
OB gene.
mechanism. Yeah.
So, yeah, I met with Jeff just after he had cloned the OB gene, which is the obese mutation, and was just preparing his Nature paper,
Wow.
1994.
Yeah.
And what he had done is he used another cloning vector called bacterial artificial chromosomes, or BACs.
Yeah.
And he convinced me that that was the way to go. And so we then recloned the region in BACs. And those are much better vectors because they're high copy number and they're grown in E. coli.
Yeah.
So you could get very high quality DNA from them. But then nobody knew any genes in the region.
So this was before the genomes were sequenced or
That's right.
known. So that was, yeah, because I was also, when I was trying to positionally clone one of the mutant in Arabidopsis, which is way much smaller. I have gone through this process of yak, back, and actually in Arabidopsis you can go to even smaller
Lambda
cosmids.
clones.
Yeah.
Yeah, cosmid. Yeah.
Cosmids. Yeah. But I remember those days when you get the back clones and you have to map them and make sure that it's the right back hasn't recombined. It's a huge amount of. I think at that time I was still doing southern blots with radioactivity
I see.
mapped. So I can feel the pain that your team must have gone through, because you make one mistake and you wipe out years of work.
That's right. So, yeah, so once we, had initiated the physical mapping part of this project, it became clear that no single person could do all this work.
Yeah.
And so I called together a lab meeting, and, in human genetics, all these projects required huge teams that work together.
Yeah.
And, so I convinced the lab that we have to just work together as a team, and each person is going to do a different aspect of the project. The back library, physical mapping, finding genes in different ways, which were strange. They don't exist today. One is called exon trapping,
Yeah.
and the other is called cDNA selection.
Yeah.
And each of those are big projects by themselves. And so about a dozen people in my lab agreed to work together.
Wow.
It took us 3 years, so it took more than 30 person years of effort.
Yeah.
But eventually we finished everything, and we eventually identified the clock gene. So the way we did it is ultimately we used cDNA selection to find cDNAs or messenger RNAs expressed from the region. That worked pretty well. But the real way we found all the genes was to shotgun sequence the back clones in our contig.
How many genes were in that interval?
So it turns out that the interval was about 200. It was 200,000 bases. Okay, that we sequenced.
Oh, you sequenced. Okay.
Yeah, we fully sequenced that region. And there are only about 3-4 genes in that region at the time. Okay. So one of the problems we had is we only had one mutant. Okay. And to prove that a gene is the causative gene in human genetics, you had to have 2 independent mutants.
I see.
Okay. So that means we would either have to have a knockout or another mutation in the same gene. Of course, we didn't have that. But I learned that you could make transgenic mice using back clones. And so 2 people were doing that, Nat Hines at Rockefeller and Eddie Rubin at the Lawrence Berkeley labs. And we then decided, okay, we're going to make transgenic mice from these back clones to test the clones to see which one had the gene by rescue. And we had other evidence, which I won't bother you with, genetically, that we could rescue with a wild-type clone for the R mutant allele.
Right.
And so we made 3 different transgenic lines, and one of them completely rescued the mutation.
Wow.
And I remember that lab meeting, I really remember. This work was done by Marina Antok, who was a postdoc at the time. And what we did was cross the back transgenic clone wild type to the clock mutants. And in her crosses, one of those lines completely rescued the mutant phenotype.
So that means instead of running at 28 hours, now it was back to 23.
20, right. And that back clone was 140 kilobases in size, okay? And the reason that was so important is, as you hinted before, if you make a mistake in that mapping, you could be off, okay? On the wrong side of the marker.
Because I have seen a couple of labs, they had made mistakes at that time. So I was scared to my... So I remember the day when I saw my mutant was rescued. I even clearly remember the time, date.
That's right.
So it's a huge moment.
Yeah, because we knew the gene is in this piece of DNA. So that was the huge moment when we knew we cloned the gene. And then after that, there was really only one gene.
I see.
Because the clock gene is very big. It's 90 to 100 kilobases in size. Has 24 exons.
Many of the clock genes, means the clock itself is, as you said, big. Even PAR is also...
they're all big.
They're all big.
So that was the key moment that we knew this was the gene, and then David King eventually found the point mutation.
That will do a lot of sequencing.
So we sequenced the gene, and he found the single nucleotide change.
How was that day when you saw the...
Well, that was also quite amazing to see what the mutation was, and it was a deletion of exon 19, which is why we call the mutation delta 19 now. And then at that point, I decided to announce the first time that we had cloned the gene at this symposium at the NIH, which was organized by Zach Hall.
I see,
And it had, it was all neuroscientists. And in my session before me was Eric Kandel.
Kandel, who got a Nobel Prize for learning.
Right. So I gave my talk and I revealed that we had cloned the clock mutation. And, it was very exciting. And then on the way back, within 24 hours of that seminar, I get an email from the editor of Searle, saying, "Dear Dr. Takahashi, I understand that you've cloned the clock gene. We'd be very interested in publishing your work."
Who was the editor then?
So, this is bad because he's very famous.
Yes, I know who he is. I'm also forgetting. Yes. In those days, he would like that. He actually used to keep 1 or 2 papers in every issue, kind of reserved, set aside. So he...
Right. Ben Lewin.
Ben Lewin.
Benjamin Lewin. Right. So that was wonderful. The only problem is that we'd already sent a pre-submission inquiry to Science. And the editor there was Katrina Kellner, who was a very good editor. So then I called another lab meeting, and we've been invited to submit our paper to Cell, but we have already sent it to Science. What are we going to do?
Yeah.
And they all decided they wanted to publish in Cell, not Science.
Because Science had published your original ENU mutagenesis.
That's right. Yeah.
Right.
So anyway, so I withdrew the paper from Science, or
Wow.
papers. There are two of them.
Two.
We submitted it to Cell. My recollection is they got reviewed in like 2 weeks and accepted. It was extremely fast.
Yeah. Ben Lewin used to do that.
Yeah.
He knew people who can accurately review, give all this. And in those days, you have to send the paper by mail.
Yeah, that's right.
FedEx. Yeah.
Right.
I remember those days.
So, yeah, and he gave us a cover picture and everything.
Yeah.
And in the end, I think we had like 25, 26 pages.
Yeah, it was one of the longest paper in those days.
Yeah, from those two papers. And, in retrospect, I go, Science would never have given us 25 pages for that work. So it was a good decision by the lab.
Yeah. No, I remember, I think that time there was a conference where you spoke and Steve K was in the audience and he came back and told us that, by the way, the clock gene is now cloned. I don't know exactly where he heard. And that was exciting for me because, the reason why I got into circadian rhythm at that time in 90s was to see all these mutants. That time the plant mutants, top 1, top 2, those were reported, and then the clock mutant was reported, and then in cyno, the mutants were reported, and then in flies and neurospora, those genes are already cloned. So I thought, well, this will be a good time to get into this field where there is
Right.
so many mutants, but not the mechanism was known. So when your clock mutant was cloned, it was quite personal for me too to see the mutants moving. And then within the next 3, I think that was the exciting time from mid 90s to 2003-4.
That's right.
So cloned.
Yeah. So it was interesting. The skepticism persisted the entire time until we cloned the gene.
Yeah.
Because even though we had this mouse mutant, they said, oh, it's too hard to clone the gene.
Yeah.
You better ask Eric Lander to help you clone the gene.
Yeah.
So in the end, we didn't have to ask Eric, but he provided lots of resources to help us. And then the other comment that I would hear is, well, even if you clone the gene, it might not be helpful. It might be casein kinase. Or calcium calmodulin 2.
Cam kinase 2.
Right, cam kinase 2. Right.
In those days, yeah.
Right.
Yeah.
So the day that paper came out, everything changed.
Yeah.
Whole world flipped.
Yeah.
So it was a wonderful time.
Yeah.
Yeah.
Then how's your journey into cloning the tau mutant? Because you mentioned relations to means you got inspired by tau mutant, and tell us a little bit about the hamster and the mutation, and then what inspired it.
Yeah, so the tau mutant had this spectacular phenotype, a 20-hour clock. But it was in an organism without any genetic resources, the golden hamster.
Yeah.
And so we wanted to map the location of the mutant.
Yeah.
And so Fred Davis and I actually worked on this when we were graduate students. We went around to all the pet shops and bought all these funny-looking hamsters and tested them.
Yeah.
And they all looked normal. They look like regular hamsters, even though they have all these coat color mutations.
So you actually went to pet store and bought.
Yeah, this is back in the day when we could do that.
Not anymore.
That was in Texas, yeah. And then when I was at Northwestern, Phil Lowry, who was working on this project, he did the same thing. We went and bought more hamsters, and he tested them for RFLPs to look for a genetic polymorphism.
Yeah.
Okay. And when we did that, we could not find a single RFLP difference.
So all the hamsters are genetically very similar.
Yeah, and it turns out the reason for that is they're all originate from a single capture
Really?
in 1930, and the founders of the colony were 3 littermates.
Huh.
Okay, and then all the rest of the hamsters in the U.S. and all the pet stores originate from that. All the coat colors are spontaneous
Oh,
mutations.
so all the coat colors came spontaneously in the
That's right.
next 60 years, but then
That's right.
a lot of the genes, majority of the genes remain the same.
Right. So the reason we're looking for RFLPs, which are restriction-linked fragment polymorphisms, is there is a method invented by Mike Wiegler at Cold Spring Harbor where he could subtract 2 genomes
Wow.
and find the difference products. Okay. And so we wanted a hamster strain we could cross to that had RFLPs, and then we could use this strategy. And so we couldn't find any until we found that an American, his name was Michael Murphy, had gone back to Syria and made a second capture and brought them back. And then he had worked at NIH. And so the hamsters were at the NIH colony.
But Syrian.
Yeah. And so we eventually tracked that down, and it turned out they were going to close the colony, so they sent us all the remaining hamsters.
This is like a murder mystery or something like that. You're going to chase, figure out.
So they sent us 6 males and 6 females. Unfortunately, they're old.
Yeah.
So the females wouldn't breed, but the males were still fertile.
Yeah.
And so we had to cross the males to our mutant line, and then the cross worked. And so we did an F2 cross. So the progeny are either wild type, heterozygous, or homozygous mutant, which we can determine by running wheel activity, period. That's what Phil did. And then what we did is we made 2 pools of DNA, a wild type pool and a homozygous mutant pool, from about 15 hamsters in each pool. And then we used the Wiegler method, which is called representational difference analysis. Subtracted those 2 pools. Then you get these little PCR products.
Yeah.
And then Phil tested them on southern blots. And incredibly, they were linked to the locus.
Wow.
It's like magic.
You have to be lucky.
No, the method worked.
The method worked,
Yeah.
But I guess the point is, in science, there's different pieces of these puzzle pieces, and you got to bring them together to make them work. And that's why science is so collaborative, and networking does matter to figure out.
Oh, yeah.
Like you said, means the Cold Spring Harbor method, and then the Syrian hamster in NIH. And then doing all of this, you were kind of the mastermind to bring all of this together, and it would be. So the point is, if you're just focusing on your lab and one method, it may not work. You have to look at it.
Yeah. So anyway, so that PCR fragment was like 500 base pairs.
Really?
Yeah. It would not hybridize to either mouse or human DNA.
Wow.
So we had to then use it to screen a hamster library. We screened a hamster lambda library, genomic library. We got 1 lambda clone.
Wow.
It was about 20 kb long, the insert. We sequenced that clone. There's 1 exon in there. But that exon allowed us to identify the gene. Okay. And once we mapped it to the mouse and human genome, we noted we could then look at the other genes in the region and test whether they were near in the hamster, and they were. And one of those turned out to be CK1 epsilon.
Yeah, casein kinase 1 epsilon.
Right. Casein kinase 1 epsilon. And the reason that became a candidate gene is that Michael Young had cloned a Drosophila mutation called double time, and that was the fly version of casein kinase 1. Okay, so we go in and sequence the mutant version of that, and it had a point mutation. And we could actually model where it was on the enzyme because the structure had been determined for casein kinase 1, and it was exactly in the right place. It was a binding in the binding site of the enzyme where a phosphoserine inserts.
I see.
And so, anyway, so that was the mutation. It was quite amazing.
Yeah. Was it the same time when Louis Potocek and Yingwei Fu came up with the FSPS?
Very close. Theirs was in 2000.
Yeah.
I'm trying to remember if our paper is 2000 or 2001.
Yeah, it's around
Yeah,
in
very close.
1 or 2 years time.
Right.
Means it was a beautiful story from hamster. You cloned it, and then in fly there was evidence from double time,
Right.
and then in humans,
Right.
in familial advanced sleep phase syndrome, they had this mutation
In the PER2,
yeah,
protein. Right.
that was phosphorylated by this. Yeah.
Mm-hmm.
Yeah. Then going back to the clock mutant mouse,
Mm-hmm.
although you looked at the circadian period length and later on you have looked at, the question always is, circadian clocks are so important to health, whether it's brain or body or other aspects. Then what inspired you to look at metabolism? Because there was a famous paper on this clock mutant mice and metabolism.
Yeah, so that's a paper led by Joe Bass.
Yeah.
And what we had noticed from breeding the clock mutant in Fred Turek's lab and my lab was that when the mice got older, they became obese, just on regular lab chow.
Yeah.
And so when Joe Bass joined us at Northwestern, he's an expert in diabetes and metabolism. He was immediately interested in this and said, "You have to put these on a high-fat diet." And so when that happened, the clock mutant mice became even more obese than wild type on high-fat diet.
Yeah.
And then they developed a metabolic-like syndrome, which was published in 2005.
Yeah.
And so that's sort of the beginning for us to focus on metabolism.
Yeah.
And then Joe Bass and I, have been colleagues ever since,
Yeah.
and have studied various aspects of
Yeah.
circadian biology and metabolism. It's been wonderful.
Yeah.
Yeah.
But again, it boils down to this question: if circadian clocks are keeping us healthy throughout 24 hours, what is its implication on aging? And your recent interest in calorie restriction, which is very important, means there's 100 years, nearly 100 years history of experiments showing if you reduce calorie, then you increase lifespan in mice, rats, and maybe in monkeys, at least in one study. So tell us a little bit about.
Yeah, so we became interested in aging, and that was really because of work on time-restricted feeding, in the field showing that was a very potent synchronizer for peripheral organs. And we actually thought that, if time-restricted feeding from work that you've done is beneficial for health, might it be beneficial for health span and lifespan, not just short-term metabolic changes. So there was a call for proposals from NIH to study circadian rhythms and aging. I don't know if you had one of those.
No, I didn't have, but a couple of my ex-postdocs, they applied. And, yeah.
Right. So anyway, so we applied and we actually got our
Yeah.
proposal funded. And what we found out when we wrote the proposal is when you look at the classic caloric restriction experiments done in mice from Richard Weindruck's lab in Wisconsin, they had for us and all circadian folks a very unusual feeding protocol.
Yeah.
They fed on Monday, Wednesday, and Friday morning, the mice, with an average of 30% reduction over the whole week.
Yeah.
Okay. So of course we know, mice don't skip days and they don't skip weekends.
Yeah.
And they usually don't eat in the morning either. They eat at night. So there are a couple things that are unusual in their feeding protocol. And so we argued that this should be reinvestigated. And we wanted to check time restricted feeding. But then once we got the grant, we realized, oh, this is going to be a lot of work.
Yeah.
Because you have to put the food on and then you have to take it off every day
Yeah.
for 3 years at least.
Yeah. So that's why I didn't have an IS grant, so I didn't pursue that. It's too much.
That's right. So our solution is always automation.
Yeah.
So we decided that we had to design a way of automating the feeding and recording when they eat. And so Vicky Costa Rodriguez, a talented postdoc from Argentina, started in the lab, and she took on this project. And so it took us about 2 years to come up with the final design of the feeder system and develop all the software and debug it and all that, which we did with Actimetrics, David Furster, and with Phenom Technology, Mike Wellams, who does a lot of work for me. And so the first experiment we did was just to take mice with our feeder system, record ad lib, and see when they ate. And we found what other people had found before, and that is the mice eat most of their food at night, 75%, but they still eat in the daytime, 25%. So then we shifted the mice to 30% reduction in calories, and we started the onset each day at either the beginning of the day or the beginning of the night. Okay. And what we found is as the mice got hungry, they ate all the food as soon as they could eat it, which was in about 2 hours, either at the beginning of the day or beginning of the night. Okay. So what that means is when you reduce calories, you're not just reducing the amount they eat with the same pattern. They completely change their feeding pattern and feeding behavior and go into extreme time-restricted feeding.
No, that was a beautiful paper, I remember.
With almost 22 hours of fasting. Okay, and we know that all three are beneficial. So we then could use that system to then look at longevity. And so what we did is we had those three groups. We had ad lib and then the ones that eat in 2 hours day or night. But then we were wondering whether that 2-hour eating was important. So we stretched that food out for 12 hours, day or night. And then importantly, we have a fifth group where the food is stretched out over the entire day.
I think that is the most important experiment.
That's
in caloric
correct.
Restriction field.
And so in the end, when we had done that survival experiment, the biggest surprise, I think, to the aging field is the group that ate continuously only lived 10% longer, even though they were deprived 30%. And then the mice that ate at the wrong time, which is the daytime, they live 20% longer because they had a fasting interval. But then the mice that eat at the right time, they live 35% longer. So it's a huge effect between 10% extension and 35% extension of lifespan. And the only variable is the pattern of food intake every 24 hours. There's no other change. The food's the same. We measure every pellet.
No, I always say that that's the milestone experiment combining circadian rhythm and caloric restriction, time restricted feeding. That's the ideal calorie restriction experiment should have been done many years ago. And thanks to CR field that they left it to us. No, actually, going back to your comment that we had to change kids, actually the history to time restricted feeding was we did an experiment where we were feeding mice during daytime to see whether daytime 8 hours feeding was shifting all the liver clock to daytime or some genes should be tied to the SCN. And we did that with cry, double knockout mice, wild type mice. And I think it was, it was published in PNAS. But then we're always skeptical whether the reviewers will accept the paper or not, and if they would ask for more experiments. So we had actually two groups of mice still on time-restricted feeding of normal chow. Of course, there is no caloric restriction. Daytime time-restricted feeding, nighttime time-restricted feeding, and ad libitum. And Christopher Ballmers, who did this experiment, he was a grad student, and he would change the cages during the weekday, and I would change the cages in the weekend. He actually kept the cage change for 3 years.
Whoa.
And we found a longevity difference, but then the study was not powered to look at longevity because we had only 22 or 23 mice per group. So by that time, Christopher was ready to submit his thesis and move on. So he moved on. He wanted to move on, and we said, well, instead of putting them on normal chow, which will be a longevity effect, because in short term you won't see too much difference. So then we put them on high-fat diet, time-restricted feeding, and that's the genesis of how the time-restricted feeding paper came about. But we had actually had the data for longevity, but then we didn't see, we didn't have the power. Like 22 mice longevity field would never take it.
Right.
So, but it was also not well controlled like you did, where your caloric restriction and distribution around the clock, around 24 hours, was extremely important. Yeah. No, that's a beautiful paper. But one thing I wanted to ask you on that paper. When you look at I'm always hesitant to use the word biomarker, but if you use, say, insulin, leptin, or insulin sensitivity, even cholesterol, was there any difference among these CR groups at any given time in their life?
Yeah, so, with glucose, insulin, leptin, there are clear increases with age in ad-lib fed. And also in the CR spread group, which lived the shortest.
Yeah.
But the ones that lived longer, very little difference.
Yeah, because that comes up many times, because people in the human time restricted feeding and calorie restriction, because there are a couple of papers that have come out combining time restriction with CR,
right,
or CR alone, but these are maximum 1 year study in humans. And they don't see a difference between these two groups for insulin, leptin. Of course, they're also taking healthy humans. So it always comes up, and I always go back to your paper and I say, look, even in mice, we know that these mice lived longer, but still there's not big differences in the middle of, I think you measured them after 46 weeks, 48 weeks.
Yes, 6 and 19 months of age,
Yeah.
Yeah, so I would say they're not biomarker predictors of lifespan.
Yeah.
Yeah.
And we're still trying to figure out what are the biomarker predictor. I think I would say that even in mice we don't have yet.
Yeah.
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 So what is exciting now? What is keeping you awake at night?
Yeah, so the big idea we're working on now is that the clock B-mal transcriptional network is upstream of almost every hallmark of aging pathway from classical aging studies, which have largely to do with energy metabolism. IGF-1 signaling, mTOR, AMPK, SIRT1, PGC1 alpha. Those are all under control of the clock. And we know with aging, the clock, function declines, amplitude goes down, gets more variable. And so what we're trying to do now is to boost the clock in older animals and to see if that maintains health
Yeah.
and perhaps extends lifespan. Because we know that already if you make mutations in clock or BML, the mice have shorter lifespans.
Yeah.
So we know loss of function leads to a shorter lifespan. Will gain of function
Yeah.
lead to increased lifespan and health span? And so those experiments are going on right now. They look very promising.
Yeah.
So I'm excited about that because that'll be a genetic proof of principle of our idea. We boost the clock,
Yeah.
we extend lifespan,
Yeah.
and then at the same time we're looking for small molecule activators of clock transcription to boost the clock.
Yeah.
And then maybe that can be used as an intervention to strengthen the clock, but also extend lifespan and health span.
Yeah.
So that's really what I'm excited about now.
Oh, that's exciting.
Yeah.
Thank you so much, Joe. And this is, we could have gone another couple of hours, but maybe we can come back again next year and then talk more about the increasing lifespan. Thank you so much.
Oh, thank you, Satchin, for your interest. Yeah, it's a pleasure.